All right, so for this video, we are just going to go through the paper from May-June 2022, paper 5.2, and just like any other planning and analysis paper, it's gonna be for 1 hour and 15 minutes and really is testing our planning, analysis, and evaluation skills. Okay, so you can refer to that question paper as you go along and you can check your answers. And of course, you can also refer to the marking scheme to see what are the possible points that can be earned. All right, so for this investigation, okay, we are looking at the root vegetable, the beetroot. All right, and then with this we have figure A and figure B. Okay, so with figure B showing a single cell from a beetroot tissue, and then in B we can also see that the vacuole contain the red pigment called betylene. Okay, so they've told us here that the pigment molecules are going to be too large to diffuse out of the vacuole. That means that they remain inside the vacuole. Okay, when the pieces of beetroot are soaked in a solution of calcium hypochlorite, the beetroot tissue changes from red to white, and this is caused by the calcium hypochlorite diffusing into, right? It diffuses into the beetroot cell vacuoles, causing the decoloration of the betylene pigment. So they've mentioned already here that, okay, the pigment is too big to diffuse out, but instead what happens here is that calcium hypochlorite is going to be diffusing into the vacuole. Okay, so we know that this question is essentially, okay, investigating diffusion. Okay, so that's worth taking note of. So you know what they are testing, and of course diffusion is from topic 4 of wherein we look at the movement of substances in and out of the membrane. Okay, so moving on, right? So what happened is that we have, okay, the beetroot, and then in figure 1.2 we said that we can see that there is a cube of beetroot before we soak them in the solution, and then the second image is what happens or how the beetroot would look like after you cut it into half. Okay, after you cut it into half, after soaking it with the solution of calcium hypochlorite. All right, so here, okay, we can see that it's all in red, and whereas here, okay, notice that, okay, they have labeled the width of the cube, and then here, okay, the idea of what going to be measured is the red zone, and then here the white area, that's the decolorized zone as hypochlorite diffuses into the cube. Okay, so this experiment is being done, right? So we see that the cubes of beetroot are being cut into different dimensions, okay, placed in beaker containing 10% of solution of calcium chloride. Now, worth mentioning or linking that when we're looking at different dimensions, that means this is already asking about surface area to volume ratio, or the effects of surface area to volume to the rate of diffusion. Now, okay, this is definitely what is being tested in one of our learning objectives from topic 4 when we're looking at movement or transport across membrane. Okay, moving on, they mentioned that the cubes were left for 30 minutes to allow for diffusion to occur. It was then cut into half, and the width of the tissue remaining red, or the red zone, was then measured. Now, what is being measured in experiment is normally what the dependent variable is going to be, because that's what we're measuring, that's the result. So we're taking in also that this is the dV. So a group of students investigate the effect of changing surface area to volume ratio, right? Already mentioned as well, so we sort of expected that already, on the diffusion of calcium hypochlorite into the beetroot cubes. And then the student used the cubes of different sizes and measured the width of the red zone after soaking the beetroot in calcium hypochlorite. So what is being measured? The width of the red zone. So we can almost identify already, as hinted by the question, that again repeating, this is also the dV. Okay, just the same at this point here. So the hypothesis is being suggested here, saying that the width of the red tissue remaining in the beetroot after soaking it in calcium hypochlorite is inversely proportional to the surface area to volume ratio. So what does that mean? That means that, right, as the surface area to volume ratio increases, okay, the width of the red zone decreases. Okay, so that's what inversely proportional means. And then there is going to be a question that's asking us to sketch how the curve is going to be looking like. And so to answer the first question, okay, which we have actually, I have already mentioned, okay, it's worth looking at the procedures of the experiment and making sure that we understand how the experiment is being conducted. Okay, and even though of course in the exam you're not allowed to do any highlighting, but I do suggest that maybe perhaps you could underline with your pencil some of the bits that you might need to take note of. Okay, identify the dependent variable in this investigation. So here I always advise the student to use, okay, how the measurements are being, so what are we measuring, to use the points that is mentioned in the question itself. So in this case we have here width of the tissue remaining, or in the second bit here in this paragraph we know that the students are measuring what are being measured, so measuring the width of the red zone. So that is how you can actually write down in terms of what is the dependent variable. Just use what the question how the question describes it. So width of the red zone. Okay, hopefully that is straightforward. The next one, sketch a curve on figure 1.3 to show how you expect the width of red tissue to change as the surface area to volume ratio changes if the hypothesis is correct. And going back, we can see that this is the hypothesis saying that there is an inverse proportion, right, to the surface area to volume ratio, meaning as the surface area to volume ratio increases, okay, as SA to volume increases, okay, the red area decreases. Now, looking at this question, you're also required to label the x's. So that's why we have 2 points here. All right. So what I have done here, I have actually sketched the graph because they ask us to sketch a curve. Now, very, very important to read what type of graph that they're asking. So since this one is asking for a curve, so of course, hoping that you take note of that, and we draw the curve. And now let's put in the labels. So according to what I normally remind the students, okay, we have the x-axis and the y-axis. So we have the x and the y. Okay, the y-axis is always going to be the dependent variable. So I'm just going to put that in brackets. So this is the IV, y is the dV. Okay, so label accordingly. We know what's the dV is. Okay, so dV is the width. dV is the width of the red zone. Okay, whereas the independent variable, which is going to be in the x-axis, that would be the surface area to volume ratio. So that's the points. The 2 points is, of course, one is asking you to label, make sure you got both labels correct, and then you got the independent and dependent variable in the correct axis, and now we have sketched a curve. So that's an inversely proportional, meaning as we increase the surface area to volume ratio, the width of the red zone decreases. Okay. Moving on. So the next bit, right? Okay, B part 1, asking us to describe. Now make it into a good habit to highlight the command words in all of the questions, right? So describe a method the students could use to collect the data needed to test their hypothesis. And the students were provided with the beetroot tissue, okay, 10% of calcium hypochlorite solution, and a standard laboratory equipment. And your method should be set out in a logical order and be detailed enough to let another person to follow. All right, so what I have done here at the site, okay, I have laid out, right, a sort of like the steps that I advise. I remind the students to always follow to ensure that they have included all the points that they might need whenever they're asked to answer such design question, planning and design question. Now, here, okay, I said that, oh, there is state the hypothesis, but in this case, we don't have to state the hypothesis anymore because the hypothesis has already been included in the question here. So we can already almost ignore that one. So you don't have to state it anymore because it's already part of the question. And then point number 2, the idea if you know what is the independent variable and how to change the independent variable. So what I've done in my answer is like according to the numbered points that I put here and their respective description, I also put the number here so you know which description matches which steps, okay, that I've included on the left side here for your guide. Okay, so the independent variable, we know that it is surface area to volume ratio, right? So what we're doing is going to be describing how the surface area to volume ratio, different surface area to volume ratio is prepared. Okay, again, we have to be able to describe them in a logical way, detailed enough to let another person follow it. So what I've done here, what we can do is cut 5 different dimensions of the beetroot. Okay, again, how do you change it? So what are the things that you need to do this? Okay, so what do we need? We need to cut it. So we're going to cut with a scalpel. And how are you going to measure the different dimensions? Of course, using a ruler. All right, and then of course, okay, we also need to remember there are things that needs to be standardized, right? Okay, we have to keep our methods the same. Now, if we look at the question, like the description in the question, notice that the width at the start, okay, there's a specific width at the start that we should keep the same because we want to be measuring how this width from the start and how the decolorization occurs. So that means we need to make sure that at least the width of the cube is kept the same for all the other dimensions. So what you probably need to do is maybe just cutting it to half and more thinner dimensions. Okay. So here, right, why did I also mention the word? So I put a very specific number here, 5. Now, I always say this to the students that, you know, make sure you remember 5 as the magic number, okay? Whenever you are asked to provide a range of any ranges of the independent variable that you want to be measuring, for example, if you're going to measure effects of temperature, that means you can specifically set, use 5 different temperature, and you mention what are the temperatures that you are going to include. So mention the 5 different temperatures and make sure if, for example, in temperature, they are written in an equal range. So 5, for example, 5 degrees, 10, 20, 30. All right, so in a logical, okay, and equal range, equally distributed range. But going back to this question, okay, we know that what we need to do is we're going to be preparing different surface area to volume ratio. What you will need to do is after you've described how it's being cut, after you've described how it is being cut, you then need to mention how you're going to calculate the surface area and volume ratio for each dimension, and we just prepare a table to record that. And of course, an example of a table would be, okay, let me just see if I could add a page here. I would like to use a dotted paper. So for example, if you have a table, right, I'm just gonna draw it like roughly. Okay. Right. right? Okay. Okay. In case of what we can do, like for example, if this was your paper 3, okay, paper 3, okay, what you can do, okay, you would put that the independent variable would be in the first column. So here you will have the surface area to volume ratio, and you state the ranges. So say 1, 2, 3, 4, 5, for example. And of course, what you're going to be doing here is you're going to be including the values, the actual calculated surface area to volume ratios. And then this is, so the second column will always have to be your dependent variable. So in this case would be the width. Okay, in case, for example, if this was part of your practical paper 3, I'm just trying to remind the students on how you should prepare your table. So remember, the first column is always have to be the independent variable. And then the second column would be the dependent variable. And of course, if there's enough time, it's always going to be a good idea to have several repeats. And whenever we include a repeat, so repeat minimum, if you have time, would be 3. Okay, repeat 1, 2, 3. And of course, you're going to have to prepare another column where you calculate the average, average or the mean. Okay, so just add another table there. I know that's like a very pruling table, but you get the idea. All right. So going back to the question, okay, moving on. So next, okay, we also were given 10% calcium hypochlorite solution. Okay, now making sure that this is also standardized. So how you're going to describe how you make sure that this is the same is that the idea of using the same volume of 10% calcium hypochlorite solution for each dimension, and you pour them in a proper container. Or you can even improve that by mentioning that, oh, maybe put them in a beaker, for example. That would be a better idea as well, as another way to improve this answer. All right, okay. So, and then remember volume, okay? Avoid to use, like, I know that this is like a bit of a habit. Sometimes students always use this word, which is amount, okay? Don't use amount. Okay, this is not good. This is very, very vague. So use the same volume. Even better if you can state the volume, if it's possible. Okay, now the next thing is, okay, we need to ensure that, okay, look at number 4 and number 5 here. We're going to look at how the steps are going to be done in a logical sequence and what are the other standardized variables. So we've mentioned, okay, the first variable is the width. Okay, the next one is we standardize the volume of 10% hypochlorite solution we're going to be using. Okay, next thing is the idea of how we are going to make sure that the procedure are the same. So what we do is here, okay, for example, continuing the description, we're going to be putting each piece of the cut beetroot earlier into the container / beaker, all right, if you want to improve this answer, containing that calcium hypochlorite solution, making sure that they are fully submerged. Now, this is very, very important, especially when we're looking at diffusion. Why? Why do you think? Why is it that we have to make sure that each piece is fully submerged to allow for diffusion to occur through all the surface? Right? This is very, very important. This is why we have to make sure that they are fully submerged. Second, okay, you make sure that the beetroot are not at the sides. They don't touch the sides or the edge of the container, because we want all of the surfaces to be fully submerged. And then we mentioned how long we are going to be submerging them. So here for 5 minutes, for example. Okay, because now we're going to be measuring, right? So after 5 minutes, okay, we take it out, we remove each of the piece, okay? The idea of how we're going to be drying up a little bit, okay, we blot the tissue, okay? Then we're going to be cutting it into half so that we can measure. So what do we use to measure? So describe it again. as clear as possible. We're going to be using a ruler to measure the width of the red area with a ruler, and we're going to be record that in your table. Okay, and then 6, there is always going to be a point for reliability. Whenever there's the word reliability, it's always like a mark scheme, control copy, control V answer, cut and paste. Okay, the description is always going to be the same. You repeat the measurement, okay, for each dimension at least 3 times, and you calculate the mean. I'll apologize for the the measurement, spelling error there. So dimension, okay, for each dimension at least 3 times. Okay, remember this magic number or formula, and we calculate the mean. Now you have to mention and calculating the mean. If you just mention repeat without mentioning having to calculate the mean, you will actually not earn that point. So please remember. And last but not least, okay, any risk assessment and the proper mitigation method. What do you mean by mitigation method? What are the procedures that you're going to do to reduce the risk? So here, okay, what I've done is an example. Okay, whenever we have chemicals or solutions, right, it will be going to be between medium or high risk. Where does this risk come from? So mention the risk. It's medium risk from where? From the solution, calcium hypochlorite solution. Why? As it may cause irritation. So that's just mentioning the risk and how, what are the dangers that it causes. Next, we have to mention the mitigation. So what can we do? We handle the chemicals with gloves, or if you don't want it to get into your eyes, we wear goggles. All right, so that's an example. Okay, so we're gonna pause there for a bit. Again, I'll let you continue with the second part so you can have a break.